Everyone, and welcome back to Human Biology here at Chaminade University. Today we're going to take on Unit 1B, talking about biological molecules and their role in nutrition. And we'd be remiss if we didn't start with water. It's the most common biological molecule in our body. All living things are comprised of water. In infants, they're about 75% water. As we get older, we get less and less, but we still end up with about 60 to 55% water in our body as we age. In males, it's about 60%. Ladies, it's about 55%. And that's because we have a different percentage of body fat. And the higher the amount of fat in your body, the little bit less water, which is why obese people actually are going to have less water in their body, as low as about 45% water, depending on their different types of BMIs, right, and their nutritional conditions. All right, so to just give you an example, a male normally is going to have about 42 liters of water in their body compartments. And we are going to be cycling through this water constantly as we are, for example, filtering blood through our kidneys and making urine, or putting things through our liver and taking out toxins, or running things through our lymphatic system as it goes back and forth between our circulation and lymphatic systems. But this water is generally going to be split up into intracellular regions, extracellular fluid, and the extracellular fluid is going to be split up into the interstitial fluid as well as the blood supply. So intracellular fluid would be fluid inside our cells. Extracellular fluid would be fluid that bathes the outside of our cells. That's the interstitial fluid, and also blood, which is going to be obviously circulating around in our circulatory system. So water is really important. All right, so let's take a look at the actual structure of a water molecule. Water is H2O. You can see here is a little upside down Mickey Mouse. So we have 2 hydrogen molecules and 1 oxygen molecule. And the reason that it's what we call polar, or going to have abnormal charges on it, is because oxygen is a larger molecule than hydrogen. Hydrogen only has that one set of electrons, so the valence shell is actually the only shell that it has. Whereas oxygen has that inner shell that's comprised of 2, as well as an outer shell that it's going to satisfy to try to get to 8. It has 6 to begin with, but it's going to borrow 1 from this hydrogen and 1 from that hydrogen. And those electron sharing is going to be how we're actually making the bonding. It's called a covalent bond between the oxygen and the hydrogen, but it's also going to infer a little bit of negative charge to the oxygen, because even though it's sharing the electrons take longer to go around this larger shell than they do around the smaller shells, right? Picture if you were running around a track that was this long, it would take you longer than this one, which means that oxygen spends a little bit longer hoarding those electrons, which gives it a little bit of a negative charge. That's depicted here by a partial. So this is partial negative. That also is going to be reflected in the hydrogens. They're going to have a partial positive charges. So although they're very well bonded together between this hydrogen and this oxygen, and that hydrogen and that oxygen, they're also what we call promiscuous in that they're also looking for other electrical charges. So 2 water molecules next to each other, for example, might form a hydrogen bond where this water molecule's partial positive charges on the H would align next to the partial negative charges on the oxygen of the water molecule next to it. Okay, so the oxygen and the hydrogen within the same water molecule are going to form a covalent bond. And the hydrogen that might be next to the water molecule here that has an oxygen, that can form what's called a hydrogen bond. That's going to be less of a bond because you've got a partial charge to give and not a strong charge, but it still can be very, very strong, and it gives the water what we call a cohesive property. All right, so as I mentioned, water molecules are going to have a slight charge on one side. 2 sides are going to have a slight positive charge, and one side is going to have a slight negative charge. That's going to be called a polar molecule. Polar molecules are hydrophilic. That means that they interact well with water. Water-loving, right? Hydrophilic, water-loving. Now, molecules that are evenly dispersed and don't have partial positive and partial negative charges are going to be called nonpolar molecules. These are hydrophobic or water-fearing. And they include things like oils and lipids and waxes, right? Fatty acids. Okay, so here's what I was talking about, how water can form hydrogen bonds with other molecules. So this is one water molecule. Again, we have a partial positive here and here, partial negative there. And that partial negative of this oxygen can form a partial bond with the partial positive of that hydrogen. And same with this hydrogen and that oxygen, and this oxygen and that hydrogen, etc. And that allows us to have this linkage between all of these water molecules, which means that when the drop comes out, it's not just one molecule. It's going to pull several things through it, right? And that's also going to allow for what we call the capillary action of water, which allows water to go all the way from the roots of a tree all the way up to the top and just be dispersed out into the leaves, right? Because each water molecule, as it goes up the trunk, it's going to pull on the water molecule beneath it and help yank it up inside the structure of the tree. Excuse me, but I digress. All right, so this also means that water is going to be what we call the universal solvent. What do I mean by that? I mean that something like sodium chloride, now this is an ionic bond between sodium, positively charged, and chlorine, negatively charged, making sodium chloride. And this bond can be disrupted fairly easily with several water molecules, because this is kind of like a dance partner that we can push one from one direction or the other direction. So several of these partial positive charges are going to add up to a negative charge, which means that we can dissociate the chlorine away from the sodium and make it be happy. It's appeased by all these partial positive charges. Same thing with the sodium. It's appeased because it's facing all these negative charges, so it's positive, and it has all these partial negative charges coming from the oxygen. So you can see the oxygen's orienting towards the sodium, all the hydrogens are orienting towards the chlorine, and in that way we can dissociate sodium and chloride very easily in water. So that's what I mean when I say that water is a universal solvent. Most things that are ionic bonds are going to dissolve immediately in aqueous solution or water-based solution. And that's why almost all of the biomolecules in our body are actually going to be held together by covalent bonds, because ionic bonds are too easily disrupted when water is present and all of this fluid is found in our body. So covalent bonds are really what we're going to be talking about when we're talking about these biomolecules that are going to be able to survive in aqueous solutions. All right, so this one here is showing a molecule that is amphipathic, and I know I just introduced a new term. Amphipathic, let me go back one, is a molecule that has regions that are polar or hydrophilic or water-loving, and other regions that are nonpolar or hydrophobic or water-fearing. And what does that mean? That means that it can reach across the aisle and interact or make polar molecules interact with nonpolar molecules. This is what happens, for example, when you have grease that's in your dishes and you add soap to it. Soap is an amphipathic molecule. Amphipathic molecules are going to be able to reach across the aisle, so it's going to bond both to water and to the grease and pull the grease off and allow the water, the greasy water, if you will, to go down the drain and take it off of your dishes. Okay, so an amphipathic molecule is going to have a region that is polar and a region that is nonpolar. And this here is actually going to be a phospholipid, which is something that we're going to see in a little bit when we're talking about the cell membrane. Remember, the cell membrane is going to keep the inside of the cell separate from the outside of the cell. And it's going to be comprised almost entirely of these lipids, it's called a lipid bilayer. So they're all going to be oriented with their heads in one direction, these polar heads, and their tails in the other direction, and that's going to be one layer. And then we're going to have an opposite layer that's going to have that nonpolar tail and then a polar head, and we'll see that when we get to the actual phospholipid bilayer. But this here is a phospholipid. Again, it's an amphipathic molecule that has a region that's polar and can interact with other polar molecules, and a region that's nonpolar and can interact with nonpolar molecules. All right, so let's talk about macromolecules, specifically macromolecules that we're going to need to utilize inside our body. Those are called biomolecules. But let's focus on a couple things first. So organic molecules are all molecules that are going to be formed or utilized by living organisms. And living things are going to be carbon-based. Okay, they're carbon-based because carbon is able to make 4 bonds. That means it can be fortunate enough to make 4 single bonds, or 1 single bond and 1 triple bond, or more likely than not, a couple of like 1 double bond and 2 single bonds or something like that, which allows us to make cool little rings and chains and all of these different structures that allow us to be able to create what we call, again, biomolecules. So the 4 major atoms that are going to be utilized to comprise all of the different biomolecules that make up living things are going to be carbon, oxygen, hydrogen, and nitrogen. And again, the building material is going to be called macromolecules. They're going to be very large in comparison to some of the other molecules, particularly things like ions that are much smaller. And the 4 major classes or types of macromolecules are going to include proteins, nucleic acids, which is like DNA and RNA, carbohydrates, which is going to include your sugars and simple sugars, and your lipids and fats and waxes. All right. all of these macromolecules are assembled from smaller, what we call monomers or building blocks. I like to refer to them like train cars. When you're making a long train, you're going to use a lot of different train cars. Each individual train car is a monomer, and when you put a couple of them together, you make a dimer and then a trimer, and eventually 3 or more becomes a polymer, right? So an assembled chain of monomers is known as a polymer, and they are all assembled the same way. Basically, you're going to start with 2 monomers or 2 or more monomers, and they're going to be linked together in a chain using something that's called dehydration synthesis. All right, so here we have 2. These are actually amino acids, not a nice of acids, amino acids. I'm just kidding. Anyways, this is the amino acid glycine and the amino acid alanine. Now, these guys are 2 separate amino acids at this point, but we're about to link them together, and the first thing that we have to do is remove a water molecule. So we're going to take OH from this molecule and H from that molecule. And that's going to, by removing these molecules, these guys come together to make water, obviously, right? But it's also going to mean that we have kind of left sticky ends on this side and that side. Picture that if I wanted to make 2 individuals hold hands at a movie theater, I might have to first remove the popcorn from one person's hand and the soda from the other person's hand, right? And then their hands would be freed up to be able to touch each other. So that's what happens here. So if we take off the popcorn and the soda, now these guys have these sticky ends left over, which are able to bond to one another to make, in this case, a dipeptide, which would be 2. If we put 3 together, it would be a tripeptide. And this is a specific type, let me go backwards, a specific type of polymer. So in this case, the monomer is going to be 1 amino acid. 2 together is going to be a dipeptide, then a tripeptide. And so the polymer is going to be called a peptide chain. All right, so again, first what happens is a covalent bond is formed by removing a hydroxyl group on this side from one subunit and the hydrogen group from this side. Again, that's the popcorn and the soda. And then that means that we're going to have these 2 subunits available for linkage through the process of dehydration synthesis. And it's called dehydration synthesis because somehow when you put the popcorn and the soda together, you get a molecule of water, right? So this is like a OH group and an H are individual, and then eventually they're going to make water, right? And we can also break down larger molecules into smaller molecules. So say we had 2, that's the dipeptide that we just saw a second ago, and we wanted to break them down into 2 individual ones. This is a dipeptide, we want to make 2 individual amino acids. In order to first separate them out, we have to break down at this right here, right? We're going to separate out. So I'm going to separate those hands that I've just put together. Then I'm going to put the popcorn in one and the soda in the other. So we've removed those sticky ends. And we're going to do that by breaking down a water molecule. That's why it's called hydrolysis or hydrolysis, right? It said hydrolysis, but I'm just spelling it out for you because hydro means water and lysis means breaking. So hydrolysis means breaking down a water molecule, poop, and then using it to pop one on this side and on that side to cap off what we call previously those sticky ends to make sure that each of these amino acids is happy. So macromolecules can be built by a process called dehydration synthesis, where a water molecule is going to be produced because each of these is going to come together to make water, and that's going to form a covalent bond. This here is called a peptide bond because it's between 2 amino acids, but we could also see this with sugars becoming carbohydrates, etc. And the process of hydrolysis is when we're going to break a water molecule in half and use it to cap off the sticky ends after we break down. In this case, the peptide bond is going to be dissolved, and we'll have the popcorn and the soda put into each of the people's hands so that we can separate back out into 2 of what we were calling monomers. Okay, so that's the process of disassembling proteins back into their components. It's the reverse of dehydration synthesis, where we take a molecule of water and break it down and add it to create 2 covalent bonds, one between this one and one between that one, and that's going to be again known as hydrolysis, or the splitting of the water molecule. Okay, so while we were just talking about that particular set of macromolecules, let's talk about proteins. So proteins are macromolecules that, again, these are polymer or a long set of train cars that are made of trains or subunits, again, monomers. In this case, the monomers are amino acids. And when we take those and put them together, again, we're going to assemble them through the process of dehydration synthesis. So when we take 2 amino acids and put them together through dehydration synthesis, we're going to create a peptide bond. And after we put 2 together, that's going to be called a dipeptide. 3 is a tripeptide, etc. The full assembled polymer is going to be called a polypeptide. Now, a polypeptide chain is just the very first step in making a protein, right? So the polypeptide chain is just going to be the actual order of the amino acids that are put together through these peptide bonds to make a specific sequence of amino acids. But we have to fold that polypeptide into a protein, Okay, so proteins are macromolecules. They're comprised of amino acids, and the order of the amino acids in the polypeptide sequence is going to determine this final protein structure. And as you'll hear me say a zillion times throughout the rest of the semester, form defines function. So the shape of the protein is very specific to the action that the protein is going to, or the role that the protein is going to play in the cell. And so the specific order of the amino acids in the polypeptide chain is going to determine the structure and therefore the function of that protein. All right, so the amino acids are going to be the monomer, the small molecule of a simple repetitive structure, which is going to have 3 major groups added to 1 carbon atom. The first 2 are going to be the same in every amino acid. That's that amino group here and the carboxylic group. And I'm going to go back a second and show you that example that we were talking about, right? Here is the amino group, and here's the carboxylic group. And when we make the peptide bond, we have the COO bonding directly to the N. Okay, so I'm just going to go forward again here and talk about that amino group hanging off one side, carboxyl group hanging off the other side of the carbon, and that third section is a functional group. The functional group is known as R. It's going to have multiple different sizes, charges, shapes, etc. It's a little dangly region that hangs off of the amino acid that gives it its specific chemical identity. Now, there are 20 different types of amino acids, and I want to tell you right now, you will not be tested on the specifics, so do not memorize this image. Just look at the fact that all of them have the carbon in the middle, all 20 of them. They all have this amino group dangling off one side, the carboxylic group dangling off the other, hydrogen, and then a structure that is different or unique that gives the amino acid its own unique characteristic, and that's called the R group. Some of them can be charged, positively charged, negatively charged, right? Acids, etc. They can be uncharged. Some of them can be polar. Some of them can be non-polar, right? The only ones that can be charged are going to be the polar ones. The non-polar ones aren't going to be able to hold a charge. Some of them are going to be very large, like tryptophan. Some of them have weird bends to them, like proline is going to cause a kink in the chain because it just can't bond properly to the one next to them. It's the only one you'll see that has a weird dangle, right? Everybody else dangles like this. This one is going to cause, as you might imagine, trying to put this in a necklace, it would cause a weird kink in the chain. So this is going to cause a turn. So all of these have their own, this is the very smallest one, they all have their own structural identity that lends to the conformation or the shape of the final protein. And the shape, again, form, is going to define the function of that final protein. So its role in the cell is going to be directly related to which of these amino acids get placed where in that developing polypeptide chain. All right, so these are different types of proteins that are found in the human body. And we're going to go over their different types as well as the general function and then just give you a couple examples of each. So when I say that there are a couple different enzymes, there are thousands, probably 100,000 different enzymes. All right, probably tens of thousands of different enzymes. Enzymes are very specific for the reaction that they what we call catalyze. What does that mean? That means increasing the rate of the chemical reaction. So every reaction that's going to happen in our body has a percentage chance of happening naturally, and then that percentage chance is going to be drastically increased when the enzyme is present. It's kind of like a matchmaker that brings two things together to allow the conversion of one thing into another. An example of this would be lipase. Lipase is an enzyme that takes lipids and breaks them down. But they gave you another example of an enzyme because some enzymes build things up. So polymerase is an enzyme that's going to take two nucleotides and combine them together during DNA replication. So some enzymes break things down and some enzymes build things up, and these two going back and forth are going to be routinely used in the process of metabolism. But I do want to say something here, that the enzyme itself is never converted from one thing into another. While it does have what we call a conformational or a shape change that occurs during, for example, lipase will change shape when it's bound to a lipid before it breaks the lipid down. When it is finished, it will return back to its native or original conformation. So it will not be changed throughout the reaction, just like a matchmaker will remain unmarried after the wedding. All right. We also have proteins that serve for protection, such as our entire immune system, for example. These guys are involved in protecting the body from external pathogens, like microbes, or any types of cellular damage that might occur. Good examples of this are going to be antibodies, which are going to be involved in fighting infections. And something that's involved in blood clotting, a little protein called fibrinogen, which is going to help us prevent from bleeding out, right? We want to make sure that if we do have a cut on somewhere in our capillaries, that it's going to close, and we do that by this clotting factor called fibrinogen. We have proteins that are embedded in the membrane. These are basically going to be involved in communication. These are signaling molecules, or they can also be adherent. So they're structural and they can be holding together, different junctional molecules, for example. Or they can also be signaling molecules, or molecules that are going to go all the way across the cell membrane from the outside to the inside to relay information or to pump things across. A good example includes a sodium-potassium pump, where we're transporting sodium out of the cell and bringing potassium in. And then here's another one called a neurotransmitter, where calcium channels are going to take calcium into neurons, so that's going to cause the release of neurotransmitters, which is how our neurons are going to fire. We have cell recognition proteins that are going to define self versus non-self, that allow the markers on the outside of cells, basically, that allow the immune cells to recognize self versus something that's not self, like a microbe or a pathogen. We have specialty red blood cell markers that will allow us to have our blood type. These are good recognition molecules, or good examples of recognition molecules. These are going to be glycosylated proteins, or glycoproteins that are for A, B, AB, and O. And we'll talk about that when we get to blood typing. We also have specific proteins called MHC complex, or major histocompatibility proteins, that are going to basically take foreign invaders and present them to immune cells so that the immune cells can then decide on the best form of attack. We have contractile proteins. This is how you're able to move your muscles at any given moment. They're voluntary, that are involved in muscle contraction and motion. And the two major ones are actin and myosin, and don't worry, we will talk about them too. We have transporter proteins that bring things into or out of the cell. Proteins like lipoproteins are going to transport fats through the bloodstream. Hemoglobin is going to pick up oxygen and bring it throughout the body and deliver it to the cells and the tissues. We also have structural proteins that are involved in making the cytoskeleton or the extracellular matrix. And what's interesting about these is, similar to the way that your house might hold up large areas, it's going to be a region that allows adherence for different, what we call the endomembrane system, which I know we haven't gotten to yet, but all of the different organelles are held together in particular locations in the cell by these structural proteins, and they serve as meeting locations as well. Just like if you were to think that like the steps of a house serve as more than just structure to hold them up, they're actually going to have transport. People are going to come in and out. They might sit there and have their drink at sunset or whatnot, right? And so these structural proteins not only serve as adhesions molecules that allow us to hold different regions of the cells together, like the organelles in place, but they also serve as meeting locations where enzymes might be able to find their substrates, etc. Okay, we have lots of different regulatory proteins that are involved in cell signaling. Anytime that we want to have, remember we had homeostasis, then we want to make sure we maintain it. So if something gets out of our homeostatic conditions, we want to bring it back in through those negative feedback loops we talked about. And so some of the regulatory proteins that are involved in responding to those feedback loops include things like insulin, which is a hormone that's responsible for regulating blood sugar by getting it out of the bloodstream and delivering it to, say, the liver and the other cells of the body, or storing it in fat. We also have growth hormone, which is going to be responsible for dealing with your bone density and your muscle density, so increasing your bone and muscle density. And we also have regulatory proteins known as troponin and tropomyosin that are involved in muscle contractility and contraction. The overview here is that there are a ton of different proteins, and they all do their very specific function based on their very specific form, and that these are very complex molecules. Protein structure is very complex. There's actually 4 different levels of protein structure. So the order that the amino acids are placed into is really important, because that's going to affect how the protein folds. The way that the protein folds is going to determine the protein's function, and sometimes proteins have more than one polypeptide linked together. Like hemoglobin is actually a quaternary structure. It's going to have 4 different proteins that link together. Okay, so there are 4 general levels to protein structure. The first we've already talked about pretty extensively. That's primary. That's the specific order of the amino acids as they're put together in this little popcorn chain, right? Then that's going to cause folding based on which regions are going to make what we call beta-pleated sheets or alpha helices. So they're going to make these specific secondary structure regions that are folded in particular ways that then make what we call the tertiary structure, which is the overall folded nature of the complete one portion or one peptide of the protein. And then multiple proteins are going to need, or multiple peptides are going to need to come together to form the final protein. Again, this is our quaternary structure when we have multiple different sets of these regions of polypeptides that come together to make this finished ultimate quaternary structure or final protein product. Okay? And again, the primary structure is going to be the amino acid sequence. Secondary structure is when we're starting to fold into what we call beta-pleated sheets or alpha helices. Why does that happen? That happens because of interactions amongst these amino acids. So let me go backwards 1 second to get to here, and we'll talk about cysteine. Cysteine is a special amino acid that has sulfur in it. It's going to allow for, methionine also has sulfur in it, but not at the end, so it's not as reactive. Okay, this one has sulfur in it at the very end. It can lose its hydrogen, and that is going to make sulfur have the sticky ends. Remember, if we talked about getting rid of the popcorn or the soda, this individual would then want to hold hands. Well, sulfur is really reactive. It's super handsy. So when that H comes off, it's going to be able to find, very specifically, another sulfur molecule. So two sulfur molecules next to each other that have had the soda and popcorn taken out of their hands are going to bond in what's called a disulfide bridge that is a form of a secondary structure that's incredibly strong. We also can have hydrogen binding, where we have the oxygen of one with a partial partial negative, and the hydrogen of one with a partial positive coming together, making a hydrogen bond. We can have hydrophobic interactions, where we have two regions that are going to not like water, and so they're going to kind of glob together the way two pieces of oil might come together on the surface of water. And then we also have ionic bonds that can happen. So there's many different interactions that occur between these functional groups that dangle off, right? These guys here are really reactive. They're teenagers at a high school dance, and they all want to interact with each other, right? And they do so by these specific chemical interactions. Then again, we have that tertiary structure, which is all of these taken together and taken into accommodation. And then the quaternary structure is when more than one protein combines to be able to make the final structure of the protein. And then here's talking about the different interactions, which I just discussed, that can cause the proteins to fold into the secondary and tertiary structures. Okay, so we just talked about the chemistry allowing for specific structures. And remember, form or structure or shape is going to affect function. And this is not just referring to proteins, this is referring to everything in macromolecules, but specifically referring to function of proteins, the shape is going to affect the function. And we can affect the shape by damaging it. Okay, so these are actually relatively fragile. And all of these things, as primary and secondary and tertiary structures, happen as the protein is being put together. So the pop beads are being put together, the popcorn's getting put together as the folding is occurring, as the quaternary structure is going together. So it's really difficult to go backwards if you were to take that whole thing and take it apart and put it back into the shape that it was originally. And so what I'm saying is that it's fragile enough that we can damage it. And if we damage it or unfold it, it's called denaturing it. And we can do this a lot of different ways. We can denature a protein by increasing the temperature too high, or changing the pH, right, too high or too low. And once we have denatured the protein, it's inactive permanently. It's not going to be able to regain its function because that folding process actually occurs as the protein is being created. The full protein is never going to be able to fold into the correct structure. So this is just showing you what happens, the denaturation, right? This is your brain on drugs. I'm just kidding. This is your protein on denaturation. So this is a native protein that has not been denatured, and after you heat it up, you can denature it. So if proteins can unfold and become denatured when exposed to heat, or also, again, that's during cooking, or also in the hydrochloric acid of the stomach. So the stomach can also serve to unfold or denature these proteins, which is one of the first steps in digestion. Okay, so proteins or food proteins are going to be natured first by cooking and then by the hydrochloric acid in the stomach. But the denaturing is not going to impact the amino acids themselves, right? Remember, we're just breaking down all of these extra things, right? We're going from here, back down to here, back down to here, back down to there. So it's not going to actually damage the amino acids because the amino acids are like the little pieces of popcorn, right, on the chain, and they're still fine during the denaturation process. Now, to be clear, while we're in the process of digestion, we are not going to be absorbing proteins across the intestinal wall. We are going to break down those proteins into their amino acids, right? We're not going to absorb the whole popcorn chain. We're only going to absorb individual pieces of popcorn and maybe small peptides, right? 2 or 3 pieces of popcorn together. All of these are the products or the breakdown of protein digestion. All right, so I talked about enzymes a little bit earlier. Enzymes are specific proteins. They're generally globular. What does globular mean? That they're in the shape of a globe, or like kind of just a ball, a glob, right? Generally, they're going to look like boogers, something like that. They have specific regions that have special 3D shape in them that are going to have a precise pocket that reacts with another biomolecule, another chemical. And this is going to be an example where form defines function very specifically. They're often referred to as like a lock and key mechanism, where although many locks look the same and all the keys look similar, only one particular key will fit into and unlock that lock. And that's because inside their active site, they're going to have specialty regions that are going to interact or fit perfectly with the substrate that they're going to catalyze. So these enzymes are going to be, enzymes are going to cause whatever chemicals that they are going to be catalyzing to fit together to undergo a reaction. Again, they are matchmakers, but they themselves do not actually ever end up married, okay? So they're going to bring molecule A and molecule B together to make a molecule that is going to, say we bring in two individual molecules and release one that is a larger molecule where they've combined. That's called a synthesis reaction. Or we can have a degradation reaction where we take a large biomolecule and break it down. So we take a protein and we break it down into amino acids, okay? So enzymes are basically going to cause these chemicals that are the precursor molecules to fit in a specific location in order to undergo a reaction, and they're used to catalyze a specific chemical reaction. Again, we have two different types of chemical reactions that can occur. We can build molecules up. These are called anabolic reactions. Picture anabolic steroids pump you up, right? Like Arnold Schwarzenegger. And then we also can have enzymatic reactions that are going to break things down. Picture like a cat on a table that's about to knock a vase over. These are called catabolic reactions, or reactions where we're breaking things down. And the sum of all of our anabolic reactions and catabolic reactions in our body is known again as metabolism. All right, so this is an example of specific enzyme function. This here is the enzyme shown in blue. This is a specific enzyme lactase, whose role is to take lactose, okay, which goes in, and break it down into glucose and galactose. It does that by having a specific region. Again, this is known as the active site. This would be where the key goes into the lock, for example. So this enzyme, which has nothing loaded into the active site, can catalyze the reaction. The substrate, lactose, is going to get loaded into this active site. And when we have these lactose molecules, then they're going to be broken down. Again, that's going to happen because of dehydration, right? Oh, sorry, that water molecule is going to, by hydrolysis, I apologize. It's going to happen by hydrolysis. We're going to break this water molecule off and cap off these two sticky ends. That's going to release glucose and galactose. And it's also going to release this enzyme completely unchanged. So the enzyme is going to catalyze the conversion of the substrates, in this case is lactose, into products, in this case galactose and glucose. They're going to be released, and then the enzyme can catalyze that reaction again. And we're just going to lather, rinse, repeat, which drastically speeds up the rate of these cellular reactions, because the chances of lactose actually breaking down into these by itself, although it still will happen, is going to happen at a much substantially slower rate. Okay. So that's proteins. Let's move on to nucleic acids. Nucleic acids are another type of our biomolecule. And we have two major types of nucleic acids, both of which you should be familiar with. One is called DNA, or deoxyribonucleic acid. The other is called RNA, or ribonucleic acid. And these two differ in the fact that they are either lacking one oxygen or have the oxygen, right? Deoxy versus having it. So that's going to be one major way that we're going to differ. Basically, we have a different sugar, right? Ribose is going to be the sugar for ribonucleic acid, and deoxyribose, which again is lacking that oxygen, is going to be the sugar for DNA. Another thing that's going to be a major difference between these two is that DNA is always double-stranded, or almost always double-stranded, unless it's being opened to be copied or to have RNA made. And RNA is always going to be single-stranded. DNA is going to stay in the nucleus. It's never allowed to leave. RNA is going to be allowed to leave the nucleus through the nuclear pores, and then is going to go on to find what's called ribosomes, and it's going to become the site of protein synthesis. We also have ribonucleic acid, or RNA, for what's called transfer RNA or tRNA, as well as ribosomal RNA. So RNA is a very versatile molecule. What does DNA do? DNA is going to have the blueprints for every single RNA molecule. So basically, we're going to have a really long scroll that has hundreds of thousands of individual genes on it, which are going to have a blueprint for an RNA molecule that codes for a specific protein. Let me give you an analogy that I like to work with. Okay, so we're going to talk about recipes for a second. All right, so if you were to figure that there are a ton of different recipes for, let's say, apple pie, right? Some people might put cinnamon in there, they might put nutmeg in there, they might use green apples or red apples, but they're all going to be making apple pie, right? But in the end, all the apple pies can be slightly different. Those are called variants, right? All right, so let's picture that the DNA is going to be the actual cookbook that recipe came from. Every grandma has their cookbook, right? So it's going to be from grandma's cookbook. It has all of grandma's recipes in it. That's the entire genome. That's the DNA. And then one single recipe that's going to be copied out of grandma's book is the RNA. But you can't eat the RNA, right? Just like you can't eat the recipe. You have to put all the ingredients together and actually make the pie. That's the protein, okay? So the DNA is the recipe book. The RNA, so this is going to be the actual recipe in the recipe book, is referred to as a gene. It's going to be copied into an RNA, which can actually leave the kitchen or leave the nucleus and go out into create a particular protein or the pie itself. And we'll see where that pie example leads us in just a moment when we talk about the difference in different types of alleles, etc. But let's focus on nucleic acids. So nucleic acids are going to be found, here's a DNA. DNA is found inside the nucleus, right? And DNA is going to be comprised of 4 different sets of nucleic acids, A, T, C, and G. And then we're going to be putting them together into what we have here is a double helix. Helix being is like a spiral staircase. And if you were to think of this as a spiral staircase, the rungs of the staircase would actually be held together by hydrogen bonds among different base pairs, A binding with T and C binding with G. And then the outer part, where you might put your hands to walk up, for example, and that's going to be comprised of the sugar phosphate, sugar phosphate, sugar phosphate backbone. And that's going to be the same on this side and the other, although this is going one direction and that's going the other. So you can see 3' to 5' on one side, 5' to 3' on the other. What does that mean? Basically, it means that here we're going sugar phosphate, and here we're going phosphate, sugar, right? So very similar, just going the opposite directions. In a minute, we'll talk about the difference between G and C and A and T, but I want to take a time right now to show you that G and C are held together by 3 bonds, and A and T are held together by 2. A really easy way to remember which of these bind together is that A and T are both comprised of straight lines, and G and C are both curved lines, so it's easy to remember that G and C bind and that A and T binds. All right. So DNA in the cell is going to be wound very tightly around things called histones. And then further contracted, if you were to picture that you're super coiling a rope, like you're twisting and twisting and twisting, it's going to finally coil in among itself. That's going to make what's called a chromosome. So again, the chromosome is the entire family recipe book of all the recipes that grandma ever made. But a particular region of it is a gene, like this section right here would be called a locus, and it would have a gene at it. And that gene would be the specific recipe for grandma's apple pie, right? Not all of her recipes, just her apple pie recipe. And everybody's grandma recipe for apple pie is a little bit different, and those are called allelic variations in that particular gene. Okay, so DNA and RNA are both going to be made of, just like we spoke previously, we've got monomers that make polymers. So the monomer of DNA and RNA are going to be nucleotides. Nucleotides are comprised of 3 major parts. First, we have the sugar. The sugar is either going to be ribose or deoxyribose. Only difference being the deoxyribose has 1 less oxygen present than ribose. We also are going to have the phosphate group. It's going to look exactly the same on both of them. And the nitrogenous base, again, that's going to look, again, the same on both of them. We have here thymine, which is something that's going to be found with DNA. So here we have deoxyribose connected to thymine, and on the other side, phosphate, and that's going to be considered 1 nucleotide. All right, so this 1 nucleotide can have different nitrogenous bases. So we have 4 major nitrogenous bases that are used in DNA and 4 major nitrogenous bases that are used in RNA. But T is specific for DNA, and U, or uracil, is specific for RNA. This is another way that DNA and RNA differ, is that DNA is going to use AT, C, and G, and RNA is going to use AU, C, and G. Okay, so I showed previously that we have a double helix. And now I just showed you what 1 nucleotide is. Again, 1 nucleotide is going to be consisting of 1 phosphate, 1 sugar, and 1 nitrogenous base. In this case, it's C, which is going to, or cytosine, which is going to pair with guanine, right? And A is going to bind with thymine. And the way that these bond together is 2 different ways. We have one that is by size and one that is by charge. If we note that every one of these has one that's a little bit larger, G is a little bit larger than C, A is a little bit larger than T. If we had two small, they wouldn't reach. If we had two large, they would be too big. So we have to have one small and one large, and we can only have either 3 or 2. So the combinations are specific. The large A, which has 2, with the small t that has 2, and the large G, which has 3, and the large small c, that has 3 hydrogen bonds. So in this way, by both size and charge, we're going to be able to have specific base pairing between A and T and G and C. And again, the bond that holds these guys together is going to be a hydrogen bond. And what I haven't mentioned yet is that the bond that's going to hold the sugar and the phosphates together, it's much stronger. It's called a phosphodiester bond. So these hydrogen bonds are kind of like Velcro. They're able to open up and come back together and open up and come back together to allow access to the specific sequence of nucleotides, which allows us to make RNA from DNA. And the stronger outside bond is going to hold the entire sequence together while it's open before it shuts back together. All right, so the sugar is going to be connected here directly to the base by what's called a glycosidic bond. All right, so again, hydrogen bonds, glycosidic bonds, and phosphodiester bonds. All right, so just a quick little quiz for you. If you know this particular sequence of DNA, what would be the sequence of the opposite strand? And the cool thing about DNA is that each side contains all of the information that you need to be able to put things together. Why? Because you know if you have an A, on the other side you have a T, a C gives a G. So this one side alone gives enough instructions to be able to make the opposing strand. So given the one side, what's the sequence of the opposing strand? That answer is A becomes T, C bonds to G, T bonds to A, etc. But if this opposite strand here was RNA being created from DNA, instead of seeing these T's, we would see a U. So A would bond with a U, G would bond with a C. These T's would still bind with A's because the T's would be on the DNA strand and the sense strand be in the RNA strand. But everywhere you see a T, we would have a U or a uracil instead. So again, anytime you see a T, you know you're dealing with DNA. Anytime you see a U, you know you're dealing with RNA. Okay, so RNA is shown here in red. It's a single-stranded molecule that's also made of nucleic acids. And it's going to be transcribed from a DNA molecule, which has a specific gene sequence. Okay, so remember the DNA codes for the RNA, and the RNA codes for the protein synthesis. All right, so in order to make RNA, we have a particular enzyme. Again, enzymes have specific forms, and that form gives them specific functions. This RNA polymerase's shape gives it the ability to unwind the DNA, allowing for the creation of this RNA molecule from the template strand of the DNA. And then after the RNA is made, this will fall off, the DNA will coil back up again into its regular double helix, and it will go about its merry little way. The RNA, on the other hand, will leave the nucleus. All of this happens inside the nucleus. Remember, DNA never leaves the nucleus. The RNA will be allowed to leave the nucleus because it goes through the nuclear pores and passes what I like to call the gatekeepers. The gatekeeper is going to ask the RNA 3 questions. Are you single-stranded or are you double-stranded? The answer is I'm single-stranded. The second question, are you using thymine or are you using uracil? He's going to say, sorry, I only have uracil. There's no thymine aboard. And then the next question is, are you using ribose or deoxyribose as your sugar base? And when that answer is ribose, that is going to give us all 3 of the correct answers for the RNA to be able to leave out through the nuclear pores to be able to find the ribosomes in the cytosol to start the process of translating this message into a protein. Okay, so just to compare the DNA versus the RNA, there are several major differences. Again, RNA is going to be single-stranded. DNA is going to be double-stranded. DNA utilizes A, T, C, and G, while RNA utilizes A, U, C, and G. You'll note there's no thymine, instead we've added uracil. The sugar in DNA is deoxyribose, so in RNA it's ribose. And the DNA never leaves the nucleus. It serves to store genetic information, but it's just going to be basically the vault that holds all of grandma's recipes. Whereas the RNA is actually going to be transcribed. We're going to be making Xeroxes of the recipes, essentially. And then those recipes that are coming from the vault can then transfer out into the kitchen, where they're going to be used as a template to actually make the apple pies, in this case, the proteins of whatever we're actually creating. All right? All right, so that wraps up nucleic acids. Let's move on to carbohydrates. Carbohydrates are going to play a really important role in energy production and storage. They also are going to make up the structural framework of most cells, and a lot of the biomolecules are going to be able to convert back and forth into carbohydrates. A carbohydrate is going to have a specific order of the elements carbon, hydrogen, and oxygen, a ratio of 1 to 2 to 1. So you may know of Glucose, for example, that's C6H12O6. So anytime that you can take your carbon and oxygen, you see you have the same amount, and you double them, or you add the carbon and the oxygen together, I guess, and you get the number of hydrogen, well, now you know you're dealing with the carbohydrates. And carbohydrates come in various sizes. We have small carbohydrates known as simple carbohydrates. This is when we have one or two monomers put together. So we either have a monomer or a dimer. We also have complex carbohydrates when we have polymers, again, trimers or longer. Monosaccharides when we have one monomer, just one train car. A good example of a monosaccharide is sugar, the sugar glucose. Sorry, these are all different types of sugars. Sugar glucose, again, that's C6H12O6. And disaccharides are when we're putting together two monosaccharides like sucrose. Sucrose is going to be formed when we put together glucose and fructose, making the disaccharide, again, sucrose. All right, so here are monosaccharides. Monosaccharides, again, they're just one train car. And you can tell they're sugars because they're going to be that nice carbon ring there. They're either going to be a 6-carbon ring or a 5-carbon ring. And they're all going to have slightly different chemical structures, right? But we're going to have similar atoms found in here, again, namely carbon, hydrogen, and oxygen. And we have many different carbohydrates that we have utilized in our everyday life, including sucrose, which is our table sugar. It's going to be a dimer of glucose and fructose. Lactose is another dimer comprised of glucose and galactose. Maltose is a dimer composed of glucose and glucose. So we use these in a lot of different places, right? This is milk sugars. This is maltose. We use this for making breads, etc. Table sugars, sucrose. We use these for a lot of different things. Okay, again, here are different carbohydrates. These are individual monomers. Here's glucose and fructose. When they're put together, they make a dimer. And the specific version of this monomer is called a monosaccharide. Two of them together make a dimer called a disaccharide. And again, this is going to happen by dehydration synthesis. We're going to remove the soda and the popcorn, right? Leaving the water to be comprised of those things, leaving sticky ends that are going to allow them to hold hands. Don't they look so cute? This is their little hand holding, right? It's like Valentine's Day. And then here's another example of a disaccharide. We have maltose here, and then here's lactose. And again, this is basically when we're bringing together these different monomers to make dimers, or monosaccharides coming together more specifically to make disaccharides. Okay, so I just showed you lactose. Lactose being comprised of what? Glucose and galactose, and we know that why? Because we saw it just previously when we were looking, I'm going very far back here, I know, but when we were talking about enzymes, we were looking at lactose being broken down into glucose and galactose by an enzyme called lactase. Again, anytime you see ASE, you know you're dealing with the enzyme. OSE, you're dealing with the sugar. So again, lactose breaking down in glucose and galactose, we saw it here. We also saw it here. Okay, lactose being comprised of glucose and galactose. Again, it's a disaccharide or a dimer. Okay, so what is lactose intolerance? Lactose intolerance is a very common condition that is caused by a deficiency in the ability to produce an enzyme lactase. So some people can't produce it at all. Some people create small amounts of it that work but not very well. And basically what happens under normal conditions, lactose is digested and absorbed in the small intestines. And if it's not digested and absorbed, the bacteria in the large bowel utilize it, but they run under the process of anaerobic respiration. So they work a little bit differently than our cells in that they produce gas, right? This is why we use anaerobic respiration to make, say, beer or to make bread, right? We use it to make malt balls. Why? Because it makes gas, and that makes little bubbles to give us fermentation, right? It gives us carbonation. So the bacteria in the bowel consuming lactose is not a good thing because it's going to produce excessive gas as a waste product. Right? So if you're unable to digest lactase in the normal location, again, that's going to be the small intestines, they move on to the large intestines where the bacteria there are going to encounter it and produce a bunch of methane gas as a waste product. All right, this is something that's easily treated. However, you can take exogenous forms of the enzyme lactase. It's called Lactaid. So if you are lactose intolerant and you know you're going to go to an ice cream function, you can take Lactaid in advance, which is basically going to be lactase that you put into your body that allows you to digest and absorb that lactose in the correct location in the small intestines. Incidentally, people who can't have milk can still have yogurt usually, which is interesting because yogurt has a ton of lactose in it. But yogurt comes with its own microorganisms, right? It's a probiotic. It has a ton of organisms in them, and those organisms create the enzyme lactase for you. So as long as you're eating yogurt, which has those probiotics in it, they create the lactase. That breaks down the lactose, so you're essentially eating both lactase and lactose at the same time. So it doesn't matter that you don't have it yourself. All right, I'm going to digress here, or I already digressed, I suppose. I'm going to return back on track here. Complex carbohydrates are long chains of polymers. Again, this is polysaccharides. So 1 monosaccharide, 2 disaccharides, long chains of them, polysaccharides. And these are going to be great for energy storage because they have a lot of bonds of carbon and hydrogen. And in between carbon and hydrogen, we're going to be able to store a lot of energy. And so because we have a lot of these bonds, we have a lot of energy that's potential energy essentially that can be stored to eventually be broken down by organisms to obtain their energy. All right, so here's an example of starch, glycogen, and cellulose. All of these are polymers. They're all polysaccharides. They are all comprised of sugar monomers, right? In this case, glucose monomers, all of them. They're all comprised of glucose monomers, but you see that sometimes they're going in a straight line, that's starches. Sometimes they're reaching across the aisle to hold hands with the person in the pew behind them, right? Sometimes they're holding hands in really opposite ways, like up, down, or down, up, right? In which case we're not actually able to access these bonds to digest them. So some of these are going to be much easier for our bodies to be able to digest than others. For example, starch is pretty easy to digest, to break off into individual glucose molecules. Glycogen is a little bit more difficult, but we still can convert glycogen back and forth into sugars by glycogenolysis, again, breaking glycogen down, making sugars. Or we could store it in the form, for example, if we have too much glucose in our blood supply, insulin will come into play and pull it out and store it in the form of glycogen so we can lower our blood sugars. Now this here is cellulose, and this is an interesting molecule. It is a very, very strong molecule that is almost indigestible, well, is indigestible to humans. There are particular organisms that are able to digest it, like termites, for example. And yeah, this is where I tell you my really terrible joke, but before I tell it, I will back it up with a little bit of information. So termites are able to eat wood, right? You guys know that. The reason that they're able to eat wood, whereas like we as humans would not be able to survive on wood, and everybody goes, oh, what about a beaver? But beavers don't eat wood, right? They just gnaw through it. My point being that cellulose is able to be digested by an enzyme that termites have that allow them to break down the bonds to free that glucose. All right, here's my bad joke. Ready? A termite walks into a bar and he says, "Where is the bartender?" Because he wants to eat the wood. All right, I digress, but that's just a remembrance for you that cellulose is able to be digested by termites and not by other organisms. All right, so we as humans are able to digest A and B, or starch and glycogen, into sugars that we can then utilize for energy. And we can store the sugars in our blood in the form of glycogen, or we can break down glycogen and release the sugars into our blood as necessary. So when we are fasting, we're breaking down glycogen to serve our needs. And when we have just eaten and our blood sugar spikes, we're taking that sugar and we're storing it in our liver, etc., in the form of glycogen. Okay, so we're actually able to measure how fast sugar enters our bloodstream after eating certain foods, right? And when the sugar is going to enter our bloodstream, it's going to stimulate the response of something called insulin, okay? And if we have too much insulin in the body, we can end up with insulin resistance, which means that adding insulin doesn't necessarily take the blood sugar and reduce it, or certainly doesn't reduce it enough. And that can lead to what's called type 2 diabetes, or insulin resistance. But there's also in juvenile diabetes, when they are just not creating enough insulin, or not creating any insulin, depending, and they have to take insulin and add insulin to their bodies to be able to break down or remove the blood sugars and put it into and build up this glycogen here in our liver. All right, so I digress. But my point being that we're able to measure how quickly the food increases the blood sugar levels. And certain things do it faster than others. Sugar and complex carbohydrates are going to enter your bloodstream much faster, right? Picture the sugar goes directly to the blood. Complex carbohydrates can be broken down pretty easily, like these starches, into sugars. But things like fats or proteins might take a while to be broken down or converted into sugars. So a higher glycemic index means that it's going to have a faster rate of getting into the blood supply than, say, fats or proteins. All right, so fiber is something that we also have to talk about when we're talking about carbohydrates, because it's a complex carbohydrate. But unlike the previous carbohydrates that we just talked about, we're unable to digest it in the digestive tract. So it's just going to pass right through. All right, and we have two different types of fiber. We have fiber that is soluble, and that means that it's able to become dissolved in water. It's a little bit more gelatinous. We also have insoluble fiber, which is going to be everything that's the excess stuff from plants and roughage. It's going to make up the bulk of your plant cell walls, so anything that's whole grain, for example. And insoluble fiber you can kind of consider like a broom. It's like a nice rough broom, and it helps move things through the intestinal tract, pulls things out of regions where they might be stuck for a little while. So fiber is going to be very beneficial for allowing things to move properly through the large intestines. Okay, fiber is a prebiotic, which basically is going to help feed the gut bacteria, and it's going to help add bulk to the digestive tract that allows us to stimulate bowel movements. It's a large brush that helps move things along, which is going to help prevent constipation. It's also going to be involved in fluid balance, which also helps us to prevent constipation. It's going to move fluid into our bowels to help move things through in a nice smooth fashion. And adults are recommended to consume about 25 grams of fiber a day in order to help increase this brushing, right? The broom going through is going to help reduce the risk of colon cancer, right? So the more your bowel moves, the less constipation, the more motion you got in there, the cleaner you keep it, basically, the less the risk of colon cancer. All right, so that's going to wrap up our carbohydrates. And last but not least, we're going to move on to lipids. Lipids are going to include triglycerides, phospholipids, and cholesterol. Now, phospholipids, you may remember when we talked about the phospholipid bilayer, it's going to be a membrane that's going to separate the inside of the cell from the outside of the cell. And then we also have, again, two different types of triglycerides. These and cholesterol can all be embedded in the membrane as well. Triglycerides are able to be what we call either saturated or unsaturated. And that basically means, do we have all single bonds, or are there any places where we have double bonds, which would be considered unsaturated? And I'll show you that in a second. We also have cholesterol. And sterols are going to be very important in stabilizing the membrane, as well as in heart health, etc. So we'll talk about the sterols in just a minute. But let's talk about all of the different functions of fats. So fats are really important in lots of different parts of your body. They're going to be 60% of the brain, and they're going to be super vital to brain function, learning, memory, and mood. They're really important in the cells. Again, here's a phospholipid. It's an amphipathic molecule, which on the outside is going to be water soluble. These guys are going to be fat soluble, water soluble. And that's going to make a barrier, basically an oil barrier between the inside of the cell and the outside of the cell. So fatty acids and cholesterol are going to be responsible for building this cell membrane and helping maintain the flexibility of this bilayer. In the heart, we're going to have a high demand for energy. And when we talk about it later, you'll discover that certain foods give you more bang for your buck than others. And fat is going to give you the highest bang for your buck. So fat gives you 9 calories of energy per gram, whereas the other ones give you about 4. Carbohydrates, etc., are going to give you about 4 per gram. So we're going to utilize a lot of fats in the heart because the heart's going to require a lot of energy, and it's going to give you the best bang for your buck. So it's going to be the best fuel for the heart. So 60% of the contractile energy for the heart is going to come from burning the fuel of fat. And we also need fat in the nervous tissue because fat's going to perform in the insulatory insulatory performance. So basically, my point being that we have an electrical impulse that's starting here and is ending there, right? And it's basically like having one long extension cord, the extension cord being wrapped in that orange layer, right? It's going to have regions that are going to block that electrical impulse to allow the electricity to get from here to there without leaking out. So fats are going to comprise the material that insulates and protects these nerves, isolating these electrical impulses, and also speeding up their transmission from point A to point B. Fats are found in the lungs. A molecule called a surfactant is very important in the lungs, basically making that lung fluid a little bit less viscous, allowing for the transfer of molecules from one side to the other. So lung surfactants are very important in the process of gas exchange. During digestion, Fats are very important because they're going to slow down digestion, which allows more absorption time. So picture that if you're traveling through the small intestines very quickly, you have less time to absorb things through that conveyor belt. But if you're going through it more slowly, that allows more things to be picked out of the small intestines, absorbed, broken down, etc. So fats are going to help with creating energy, so they're going to be an energy source, and also keeping the body satiated for a longer period of time. We also have specialty vitamins that are fat soluble. So just like anything, things are either fat soluble or water soluble, and vitamins are no different. So some vitamins are water soluble, some are fat soluble, and the ones that are fat soluble can only be absorbed when fat is being picked up across the digestive tract too. So we have to have fat present when the vitamins are ingested in order for the vitamins to be able to hitch a ride with that fat to go across the intestinal layer and be picked up. And then also, we use fats as an internal cushion, a layer that helps us protect our internal organs from injury and also from pulling on each other, from having friction amongst one another as you twist and turn. We also use fats in our immune system to help with metabolism and decreasing inflammation. So as you can see, fats, lipids, waxes, and oils are going to be very important in the human body in multiple different organ systems. All right. So fats are going to be comprised of two different monomers, fatty acids and glycerol. All right, and that's going to make these fatty acid chains that are known as hydrocarbon chains. A hydrocarbon chain is just hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, until it is eventually going to end in a carboxyl group, COOH, which when we saw our carboxylic acid, when we saw our amino acids. Here, we're looking at a triglyceride being 3. Right, so we have 3 fatty acids attached to a glycerol background, so triglyceride. Here's the glycerol backbone. Again, here's our fatty acid comprised of a hydrocarbon chain. Now, if all of the carbons in this hydrocarbon chain are saturated with hydrogens, meaning that there are no double bonds between the carbons, we're going to have a nice straight line here. It's a nice organized, orderly setup, right? But if, for example, let me go forward. Oh, it's not going to show me. If, for example, we have a double bond that occurs because they were missing this hydrogen and that hydrogen, allowing this carbon and that carbon to double bond, that's going to cause one region of unsaturation where we're going to have a fold. When we have a fold, we're going to have a kink in the tail. So saturated fatty acids are going to lie very nice and flat, whereas unsaturated fatty acids are going to have kinks and folds, which are going to allow them to be slippery. I like to give this akin to, this is a nicely folded paper clip. This is a paperclip after you've bent it out of shape. You can hold all 100 paper clips in that paperclip box if they're nice and flat. But if they have this odd shape, you can't hold 100 paper clips in the box because they don't fit on top of each other, right? They slip and slide all over each other. This is the difference between saturated fatty acids and unsaturated fatty acids. Saturated fatty acids, like butter, are going to be nice and tightly compacted because they're going to fit well together in the box. That means that they're going to hold their congealed state at room temperature, as opposed to something like olive oil, which is the liquid at room temperature, because of the fact that you're not able to fit as many molecules in the same amount of space because of these unsaturated connections between the carbons. So unsaturated fats are going to be liquid at room temperature, and saturated fats, again, solid. All right, so what are some examples of saturated fats or foods that contain these saturated fats? Like avocado is a good one, poultry products, meat in general, dairy products, eggs, lots of different oils though, right? As you might expect, coconut oil, palm oil, and then butters as well. And then unsaturated fats are going to be things like olive oil, fish oil, peanut oil. Picture any oil that is actually a liquid at room temperature, whereas coconut oil is going to be a solid at room temperature. You might have to heat it up. Corn oil is unsaturated, vegetable oil, canola oil, safflower oil, etc. However, these are types of oils that are going to be a lot more refined than olive oil, fish oil, and peanut oil. So while these guys are both going to be unsaturated, these ones in the top, like olive oil, fish oil, and peanut oil, are going to be considered more healthy than corn oil, vegetable oil, etc., which are considered less healthy. Okay, so let's talk about trans fats for a second. All right, when we think about cis and trans, what we're actually saying is whether or not we are on the same side or opposite sides. So if we have I'm going to go backwards, an unsaturated fatty acid, that means that the hydrogens are dangling either both on the same side, cis, or on opposite sides, trans. If they are dangling on opposite sides, you can imagine this might compact a little bit better It doesn't have as many kinks, so it's going to appear or be able to appear as flats. Even though it's unsaturated, it's going to have a much more compactable shape. That's going to be called a trans fatty acid. Now, trans fats are going to be created by hydrogenation, where we heat up vegetable oils while we add hydrogen gas. And these partially hydrogenated vegetable oils are going to be much more shelf stable and are going to have a much longer shelf life, so they're less likely to spoil over the duration of time, which is really good for food manufacturers because they can leave that Twinkie on the shelf for 50 years and still have it edible. But that doesn't mean that it's good for you, right? It's not going to be as easy for your body to digest these trans fats. And trans fats are found in many different types of cooking, things like vegetable shortenings, margarines, a lot of things that are going to be processed, like chips and cookies and crackers and fast foods, as well as anything that's going to be fried in hydrogenated vegetable oil, so fried foods, chips, etc. All right, so here's how we make trans fats. Here we have a cis fatty acid, cis being same, trans being opposite, right? So cis hanging on the same side, two hydrogens, that's going to cause a kink. This is going to be more slippery. Here's a trans fatty acid where we're going to have the H dangling on opposite sides. And these are going to be less healthy for you, but because of their structure are going to be less likely to degrade, which means that they're going to become more shelf stable. So they're often used in highly processed foods, particularly in stuff you might find at a gas station that's meant to survive a zombie apocalypse. All right, let's talk about other triglycerides, including refined oils, such as canola oil, corn oil, vegetable oil. These guys are refined by going through multiple repeated processes, which can destroy the double bonds in your unsaturated fats. Basically, the way that you refine oil is by cleaning, flaking, cooking, pressing, extracting, desolvatizing, refining, bleaching, deodorizing, and processing. So instead of just pressing the oil, which is how we make olive oil, we literally just press the olives. How we make peanut oil, we press the peanuts, right? And you just get the oil out of it. You have to take a lot of different steps to refine canola to be able to make it into oils. So processed oils or highly refined oils are found more likely in processed foods, and they've been linked to multiple different health issues. So you want to be careful of the types of oils that you're including in your body if you want to reduce your risk of heart disease, liver dysfunction, cancers, obesities, etc. Okay, so when I say saturated versus unsaturated, that's the first differentiation. But then when we're talking about unsaturated fatty acids, some of them are monounsaturated and some of them are polyunsaturated. What do I mean? Do you have one double bond or do you have many? So this would be considered a monosaturated trans fatty acid. Monosaturated cis fatty acid. If there were another double bond here, it would be a sorry, I lost myself here. Polyunsaturated fatty acid. There we go. So we have multiple different types of essential fatty acids that we need to get from our diet. Basically, if we're able to convert it from other food, it's considered non-essential. If we are only able to eat it from a particular type of food or from our diet, that's considered essential. We have multiple different types of acids that are found, or these different types of fatty acids that can be found from things like fish, tuna, salmon, mackerel, or often from dark green vegetables as well. So leafy vegetables, also nuts and flax, yada yada. These are specific types of fatty acids that are considered to be essential. The top is alpha linoleic, the bottom is linoleic, and the third is oleic. And don't worry, I am never going to ask you specifically to look at them and tell me the difference. But if I were to show you this, I would be able to ask you if this was mono or polyunsaturated. Well, first I would ask you if it was saturated or not, right? Saturated or unsaturated. You would say it was unsaturated because we have double bonds. I would say very good. Then I would say, is it monounsaturated, like this one, or polyunsaturated, di or triunsaturated, like those guys, all right? So I want you to understand how these different bonds are going to relate to whether or not a fatty acid is saturated or unsaturated. Okay, let's move on to cholesterol. All right, cholesterol has multiple different functions. The one that you might hear of often is the creation of steroid hormones like testosterone or estrogen. And it's also going to be involved in or embedded in the cell membrane, which relates to the cell membrane flexibility. The concentration of the cholesterol in the membrane directly relates to its fluidity or flexibility. We also use cholesterol in the creation of bile. Bile is going to be created by the liver, stored in the gallbladder, and released into the small intestines to help break down fats. And last but not least, cholesterol is going to serve a very important role in lipid transport by transporting fats through the bloodstream in what we call high-density lipoproteins and low-density lipoproteins. So when we talk about cholesterol, we don't just say I have high cholesterol, right? We're looking at a ratio of the HDL and the LDL, and that actually stands for high-density and low-density lipoproteins. But it's really easy to remember that this is the healthy cholesterol and this is the lousy cholesterol. So it's better to have a ratio where you have a higher number of this and a lower of that than the flip side. All right, so we find lipids in the cell membranes of all sorts of different organisms, plants, animals, fungi, etc. We are going to have cholesterol synthesized again by the liver. So the liver is going to be producing the bile, which is a major function of cholesterol. And then also the cholesterol is going to be mainly found in the liver and the brain inside the cells. Okay, so this is what cholesterol looks like. We're going to have here 3 6-carbon rings, so those are called hexamers, and then 1 5-carbon ring, that's called a pentamer. And that's basically just going to be the structure of cholesterol. So let's move on to phospholipids. Phospholipids are the fats that are found in the cell membrane. Again, cholesterol can also be embedded in the cell membrane. But phospholipids are super cool because they're amphipathic. That means they have one region that's going to have a tail region that's going to be nonpolar, or water-fearing, or again, oil-loving. And then we have a polar head, which is going to be polar, and therefore, because it contains a phosphate group, it's going to be polar, and therefore interact well with water. So it's going to be hydrophilic. Again, these are amphipathic molecules that have one nonpolar and one polar region. Here we have the hydrophilic, or polar head, and the hydrophobic, or nonpolar tail. And this phospholipid can either be completely saturated, as in a straight line, a nice unbent paperclip, and has all single bonds, or it can be unsaturated. Again, it can be monounsaturated or diunsaturated, and that's going to give it a little kink in its tail here, which is why this one has a twist to it. Now, these are all going to be oriented in what we call a cell membrane, and a cell membrane serves to separate the inside of the cell from the outside of the cell, and it's comprised of what we call a phospholipid bilayer. So each one of these, that ball and 2 legs, is going to be considered a phospholipid. And they're all going to be oriented so that all of their heads are going to be in the same direction and their tails are going to be in the same direction. And that's one half of the phospholipid bilayer. And then the other half is going to be oriented towards the center. So we can have an aqueous solution separated from an aqueous solution by a layer of oil, essentially, right? By a layer of these nonpolar tails. And that allows us to keep the outside separate from the inside so then we can maintain a little more homeostasis on the inside. So cell membranes are comprised of that phospholipid bilayer, as well as embedded with cholesterol, right, shown here, as well as multiple different proteins that serve many different purposes. They can serve as recognition proteins, identifying self as non-self. They can serve as receptors for different biomolecules, etc. So they're going to have a lot of different, or structural proteins, yada yada. Okay, so let's talk about different types of micronutrients. Micronutrients are also known as vitamins. And vitamins all serve a particular role in metabolic functions. For example, vitamin A is going to assist in night vision. And if people end up with low amounts of vitamin A, they can end up with things like pregnancy blindness, because vitamin A can end up depleted in pregnancy. Or if anyone's ever seen Schindler's List, the man ran away and had really bad night vision because of the fact that they had been starving for a while, so he didn't have enough vitamin A. And so deficiencies in vitamin A leads to night blindness and can also lead to increased infections and dry skin. And if a deficient occurs during pregnancy, we can end up with fetal deformities. So we want to make sure that this is included in our prenatal vitamins. We also have B-thiamine, found in grains and liver, that helps produce ATP and functions in nerve function and appetite. If we have a low amount of it, we can become fatigued, very weak, have a loss of appetite, and have emotional and cognitive disturbances. This is something that happens quite often in alcoholics, because alcoholics tend to become deficient in B1-thiamine because of the way that alcohol is processed. We also have riboflavin, or B2, which is also involved in ATP production as well as skin and eye health. And if we end up deficient, we can end up fatigued or with low growth or dermatitis or skin infection, as well as cataracts can form in the eyeballs. B3 is known as niacin. It's also going to be involved in ATP production as well as digestive health and skin and nervous tissue health. And if we end up with a deficiency in niacin, we can end up with something called pellagra, which is a specific disorder marked by diarrhea, dermatitis, dementia. We can also end up extremely weary, weak, have issues with digestion, skin lesions, as well as mental confusion. You become very addled once you end up tired, and that leads to emotional disturbances. Again, this is another thing that occurs in alcoholics. B5 is also involved in production of ATP, again characterized by fatigue and weakness as well as neurological symptoms. However, this is a very uncommon symptom. This is usually found in extreme starvation conditions because B5 is found in almost everything that you eat. So as long as you are getting nutrition, you're probably getting enough B5. B6 is also involved in ATP production as well as formation of hemoglobin, so really important in blood creation or blood supply as well as in the health of the skin. And again, like we saw previously, it can be characterized, or deficiency can be characterized by fatigue, dermatitis, and in this case anemia, which goes right hand in hand with the formation of hemoglobin. Biotin is involved in the metabolism of fats, proteins, and sugars, and its deficiency is characterized by mood swings and fatigue. Again, it's also pretty uncommon because biotin is found in almost everything. So again, you have to be in pretty severe starvation conditions. It can be found in anorexics, for example. B12 or cobalamin is involved in DNA synthesis and red blood cell production. So again, you're going to see anemia if you're deficient, as well as weakness and fatigue. But we're also going to see, because it's involved in the nervous system, numbness and tingling or other neurological degradations, as well as emotional swings and mood problems. And this can often happen from lack of absorption. So it's not necessarily going to happen that you're not ingesting as much, but if you have a deficiency in absorption because you're not making enough of the IFs or intrinsic factors, you can end up appearing as though you have deficiencies. Folic acid is really important in metabolic functions of proteins, as well as the creation of red blood cells and cell growth and differentiation. And this is something that is highly pushed in your prenatal vitamins because it can cause neural tube defects like spina bifida in the developing embryo, as well as in the mom, anemia, and fatigue. We have more micronutrients like vitamin C, D, and E. C is involved in collagen production and is involved in wound healing and immune function. Also acts as an antioxidant and increases your ability to absorb iron. A deficiency in vitamin C creates scurvy, which is defined, which is something that actually hasn't happened since pirates, right? Usually if you were out at a boat at sea for a long period of time and you weren't able to get fresh fruit, you would end up with a collagen loss, which can cause your teeth to become loose in the socket. Also, wounds would end up not healing very well. You'd end up with bleeding. Infections can take root when you have poor wound healing, as well as bruising, because you're going to have bad wound healing internally as well, and bad tissue health. And it's also a common deficiency these days in heavy smokers and extreme alcoholics. Vitamin D is involved in calcium regulation, which, as you might imagine, is also involved in bone density and immune health. And if we have abnormal amounts of vitamin D, we can have rickets. We could end up with increased infections, autoimmune diseases, as well as immune types of cancer or immune-related cancers. Vitamin E is an antioxidant and is involved in the immune system. And without vitamin E, we can have impaired healing of our wounds, as well as impaired immune function and nerve problems. Other mineral micronutrients, so we just looked at all the vitamins. Now we're going to switch over to minerals, not all of them, but many of the vitamins. The minerals are going to include calcium, magnesium, phosphorus, etc. Calcium is involved in teeth and bone health, as well as in nervous transmission and contraction. And we need vitamin D in order to absorb calcium in the small intestine. So calcium being present isn't the entire story. We need to have vitamin D to hold its hand as it's being absorbed. And if we have a deficiency in calcium, we can have a decrease in bone density, known as osteoporosis. Typically happens as we get elderly, particularly in women, because women, especially women who have born children, may have had their bones depleted of calcium during the process of creating those kids and might not have restored that calcium. It also, if we have a deficiency in calcium, we can have muscle issues like muscle twitching and heart palpitations, as well as mood swings and mood problems. Magnesium is a soothing chemical. It's an anti-stress chemical that helps balance out the calcium in your nervous system. It's also a cofactor for cellular enzymes. And if you end up deficient in magnesium, it can cause muscle cramping, palpitations, heart problems, basically, fatigue, memory issues, mood swings, numbness, and tingling. This is another common deficiency in alcoholics due to nutritional deficits that normally or can occur as a side effect of alcoholism. Phosphorus is really important in making up, remember that phospholipid bilayer we talked about? Yeah, the phospholipids are right in that. Also ATP, that P stands for phosphorus. Plus, we also are going to, or phosphate, right? We also are going to have it in the nucleotides. Nucleotides, ATP is is a version of nucleotide. Remember, adenosine is going to be a nucleotide, which turns into adenosine triphosphate. But A, T, C, and G are all nucleotides. Remember, that's going to have that phospho group 2 and that phosphate sugar backbone. And a deficiency in phosphorus is not really very common because there's an abundance of phosphorus in most foods. So typically, low phosphorus conditions, again, are going to happen during starvation, and that can lead to stunted growth and bone deformities. Other minerals include iron, zinc, and iodine. Iron is really important in hemoglobin. It's actually going to form the heme in hemoglobin. It's what gives blood its red color, like rust, right? Iron oxide is rust. Hemoglobin is from the iron. The red in the blood is from the iron in the hemoglobin. And that's going to be responsible for carrying the oxygen to the cells. So if we have low iron, we can result in anemia. In fact, one of the first things they do for anemics is prescribe them iron. It can also cause muscle fatigue, general fatigue, and memory and cognition problems because it's used in a lot of the neurological molecules. So you might not be able to have a reasonable memory. Zinc is involved in immune function, particularly in wound healing and repair. It's also involved in metabolism. So if we have low amounts of zinc, we can have higher levels of infection rates, lower wound healing, also fatigue, and sometimes they'll end up with little spots on the fingernails, and also it can be associated with a sense of taste because zinc is involved in the gustatory pathways. All right, iodine is found in the thyroid hormones. It's involved in the creation of the thyroid hormones. And if we don't have enough iodine, we can end up with decreased metabolism, fatigue, because remember, thyroid hormones are going to be involved in a lot of different metabolic pathways, which can lead to fatigue, weakness, and also hypo or low amounts of thyroidism, right? So we might not be making the right amount of thyroid hormones. Okay, so that's going to wrap up our chat on the micronutrients, including vitamins and minerals. And now this is the application question. Again, we're going to have one of these at the end of every lecture. This application question is talking about considering the following meals and snacks and think about which foods would contain the highest amount of proteins, nucleic acids, carbohydrates, and fats, as well as which foods would have the most fiber, which would have the most micronutrients, and which foods would contain trans fats. You're given options of a hard-boiled egg with a piece of whole grain toast and butter, a blueberry muffin, a salad with mixed greens, hemp seeds, green peppers, radish, cucumbers, and olive oil, a bag of potato chips, and a coffee with cream and sugar, chicken, white rice with legumes and asparagus, and crackers with almond butter. And this is going to be your answer to your application questions. The foods that are going to contain protein on that list are going to include eggs, hemp seeds, chicken, and almond butter. Foods that contain nucleic acids are going to include any whole food with plant or animal cells, including eggs, greens, hemp seeds, green peppers, radishes, cucumbers, rice, legumes, etc. And the simple carbohydrates are going to be found in your blueberry muffin and your coffee, which contains sugar, right? The sugar is going to be your simple carbohydrates. Your complex carbohydrates are also found in that blueberry muffin, as well as anything that has starch, like the whole grain toast, the rice, and the crackers. And foods that contain saturated fat, are going to include things like eggs, butter, and chicken. Unsaturated fats are going to be found in the blueberry muffin, olive oil, potato chips, crackers, and almond butter. And fiber is going to be found in the toast, the greens, the hemp seed, green peppers, radish, legumes, asparagus, almond butter. And some of the trans fats could be found in anything that's going to be processed, like the blueberry muffin, the potato chips, and the crackers. Thank you so much for listening to my chat today. I really appreciate your time. So aloha, and I will see you for next lecture. Have a great day.